Okay, I'm going to start this by saying I lied. I initially said I had this lecture divided into two parts, but I'm actually going to split it into three parts. So, in part one, we talked about portals of virus entry. In the second part, I'm going to talk about points 2, 3, and 4, dissemination, portals of exit, and then patterns of infection. And then in the third part, I will talk about a little bit about epidemiology. All right, so let's talk about dissemination. How does a virus move within a host? So if a virus does not move at all or moves in a very limited way, we would consider that to be a localized infection, right? It's basically only replicating in cells at the initial site of the infection. And that is contrasted with a systemic infection, which is where a virus is either spreading throughout the entire body or at least from, one infected organ to another, or something like that. Um, so how does systemic infection occur? Well, really two main mechanisms. One would be what's called neurotropic spread, where the virus is spreading through neurons. So in this case, the virus would be replicating in the local tissue, then infecting the neurons, and because neurons are very long cells, right, that could then transmit it to additional parts of the body. Hematogenous spread is spread through the bloodstream. Anytime you have virus in the bloodstream, this is considered a viremia. We'll see as we go through some of the future chapters, they'll talk about like primary versus secondary viremias. These would basically be waves of uh, large amounts of virus in the bloodstream. And so a viremia, that hematogenous spread, could occur directly if the virus gained entry, maybe through injection or through an animal bite into the bloodstream, or it could be indirectly, as we were just discussing with neurotropic spread, where again the virus replicates locally, but then just accumulates to such high amounts that it finds its way into the bloodstream. That would be more typical. And so, of course, the bloodstream is capable of carrying the virus, throughout the entire body. But I also wanted to mention the lymphatic system. If you're not familiar with with that, um, I like to think of the lymphatic system as sort of the shadow circulatory system. So you have lymphatic vessels. It's a very dense network of vessels throughout your whole body, um, and lymph nodes. And so the job of the lymphatic vessels is basically to collect fluid and blood cells that may have leaked out of capillaries and basically return that back into the bloodstream. And the lymph nodes, you have a lot of immune cells there filtering and looking for foreign particles. So virus could end up in the lymphatic system, right, either through the bloodstream initially or could become introduced into the bloodstream from the lymphatic system. But those two are definitely connected, closely connected. All right, let's talk about virus exit. And, there's lots of different portals of exit. This is basically usually called virus shedding. An infected individual sheds virus. And, this could be happening through the skin, through respiratory secretions, diarrhea, vomit, saliva, genital secretions, blood, urine. If it's in the urine, it's called a viuria, feces. I'm not going to go through all of these like we did with portals of entry. Instead, I'm just going to focus on respiratory secretions here, particularly relevant since this is thought to be the primary portal of exit for COVID-19, although it also be found in feces and saliva and genital secretions. I think that's been demonstrated as well. So if we're talking about respiratory exit, um, you know, we're usually discussing what we consider airborne viruses. So you're going to get virus expelled when somebody coughs, sneezes, speaks, or even just breathes. Um, and we can talk about, distinguish between two different types of airborne virus. So it can be present in droplets, which are larger airborne particles, about 20 microns in size. Those are only going to spread a short distance. They're heavier. They're going to fall out of the air more quickly, especially at higher humidity. You see this gross picture here of the guy sneezing, right? You can see some of these droplets basically like immediately falling out of the air, whereas aerosolized particles are going to be about 5 microns in diameter. Uh, these are going to stay airborne for a long time. So what you see here is basically the fine mist. Um, so, when we consider mask wearing for COVID-19, masks are going to be very effective at reducing the amount of droplets that might be distributed, because if you sneeze or cough, those droplets are going to end up in your mask instead of in the air or all over whatever surfaces are around you. Aerosolized particles are a little bit of a bigger problem because they do hang in the air for a longer amount of time. You could end up with aerosolized particles just from people talking or breathing, and those, may be able to find their way around masks, unless you're talking about like an N95 or something like that. But um, could still be your biggest risk, even wearing masks, would be through aerosolized particles. So, how stable virions are going to be in the environment in general, right, is going to depend on a couple of factors. Not only what is the environment that we're talking about, but also what is the virion itself. Is it enveloped, non-enveloped? What kind of capsid protein does it have? But what's the nucleic acid type, DNA or RNA? DNA tends to be more stable than RNA. But depending on the particular characteristics of the virion, you can see the list here, right, size, absorption materials, etc., and some of these common environmental factors, temperature, pH, humidity, um, enzymes that might be present in the environment. All of these things come together to affect how stable a virion might be, and that could vary widely, obviously. So it could be stable for months or years, could be stable for hours or minutes, just depending on the particular environment. All right, so let's talk a little bit about some common patterns of infection. And this is an important slide. You're going to see some of these terms coming up throughout the rest of the semester. But anytime a host becomes infected, there are these 4 key stages of disease development. So here we have virus infecting the host, and that begins the incubation period. The incubation period is going to be the period of time before any symptoms appear at all. So you're infected, but you don't know you're infected. At the end of the incubation period, you will typically, depending on the virus, tend to get nonspecific symptoms. So, I'm achy or tired or have a bit of a fever, right? However that might manifest. Those nonspecific symptoms indicate the beginning of what's called the prodromal period. At the end of that, we have the appearance of specific symptoms, and at that point we are into the illness period. And then at the end of the illness period, symptoms are subsiding, and we go into convalescence, basically that recovery period. And you'll notice here we've got a bracket, the period of communicability, which is basically the time at which the infected person is capable of easily transmitting that virus to a new host. And typically that will begin in the incubation period, so notice before you even have any nonspecific symptoms, and may extend well into the convalescence period, just depending on the particular virus. And typically the incubation and prodromal periods tend to be shorter than the illness and convalescence periods, but again, that can vary pretty dramatically from virus to virus. So this table is showing you average incubation period and period of communicability for a number of different pretty well-known viruses here. And just wanted to point out that, again, it varies quite widely, right? So, for example, rhinovirus, you've got an average incubation period of 1 to 3 days, typically only about 24 hours. Whereas something like hepatitis B, right, could be about 4 months. So that's a big range. Um, Period of communicability, again, rhinovirus, it's about 24 hours before to 5 days after symptoms happen, kind of on the shorter end. And then you've got some viruses like HIV, where it basically extends from early in the infection to forever, right? For the rest of your life, you would be capable of transmitting that virus. It's still early days with COVID-19, but we know the average incubation period is typically 2 to 14 days. Um, and then we just don't know yet period of communicability, but we do seem to be able to detect virus even weeks into the convalescence period, but it's a little bit unclear whether that detection really indicates infectivity or not. All right, so a few terms to bring up here that I'll tie in with this idea of patterns of disease. These are basically certain different types of infection. You could have inapparent, abortive, acute, persistent, slow infections. Just define these terms. So an inapparent infection is one where the host is infected, but there's no signs or symptoms, right? That seems to be the case in some percentage of people infected with COVID-19. Asymptomatic individuals, you would say. An abortive infection is where an infection is established, but it never really gains a strong foothold, so the virus is not capable of producing any new infectious particles. So you do have some infected cells, but it's not really amplifying or spreading. An acute infection, something you don't want, right? Rapid replication of the virus spread within the host and to other hosts. Persistent infection, this would be a long-term infection. Um, the host's immune system just isn't able to clear it. Could be something like herpes. They say herpes is forever. It's because the herpes virus becomes latent and basically just hides out and then reactivates for short periods of time, but your immune system is not able to clear it. Or something like HIV, where the virus is integrated into your genome. And then sort of a subset of persistent infections are what are called slow infections, which are those that progress slowly. So it takes a long time, maybe even years, to reach that symptomatic phase. And a good example of that would be HIV, right? So you get some initial sort of non-specific symptoms very early in the infection. Those go away, and then it may be, many years before you really get into the immunodeficient symptomatic phase of HIV. I will stop there, and in part 3 we'll talk a little bit about epidemiology of viral infections.